Right now we will be discussing a very important topic, that is control of genetic elements, that is control of a gene, that is how will you turn on a gene. Okay, now listen, this particular topic has two main important subheadings, that is control of genetic elements in prokaryotes and control of genetic elements in eukaryotes. Today we will be discussing control of genetic elements in prokaryotes. Clear? Now, do you know what is the important thing about the control of genetic elements in prokaryotes? The important thing about the control of genetic elements in prokaryotes is that it is known by the name of Lac Operon primarily. Okay, it is known by the name of Lac Operon primarily. Clear? That means it's a mechanism by which bacteria turn on and off particular genes according to extracellular concentration of lactose. That is, whenever bacteria senses that lactose is present, then it turns on these genes associated with lactose operon or Lac Operon, and when lactose is absent, then it turns off. Actually not turns off, because these genes are constitutively expressed. We'll talk about these things, don't worry. Okay, so today we are going to discuss Lac Operon in details. Clear? We will be discussing first the basic concept of Lac Operon. The basic concept of Lac Operon. That means how it works. Okay. Then we will talk about different mutations. Clear? Different mutations. Then we will discuss some sums associated with these mutations. Okay. So right now we will be discussing Lac Operon on the basis of three distinct parts, that is basic concept of how Lac Operon works and what are the genes associated with Lac Operon first. Second is different mutation, and third is how this different mutation leads to some of the sums which come in your exam. These are not actually sums, these are some conditions. Okay, clear? Okay, so let's talk about at first the basic concept of Lac Operon. Clear? So right now we will be discussing the basic concept of Lac Operon. Now listen, before I start Lac Operon, you must understand basic things about bacteriology and bacterial genetics. Not bacteriology, bacterial genetics. Listen, you know that if I ask you that where is Lac Operon genes present, I will talk about what is Lac Operon in details when I have finished it. Listen, what do you mean by this term operon? I will talk about this when I have discussed all these things. But before that, you must understand, or you must tell me, okay, and help me understand that where is Lac Operon genes present? Where are Lac Operon genes present? Listen, if this question confuses you, then I must tell you, you should understand this class, or you should take this class very seriously, because probably you don't know what is HFA strain. Clear? Or what is normal genetics of bacteria. Listen, suppose this is a very cute looking bacteria. Okay, this is suppose a very cute looking bacteria. Okay, now listen, do you know the speciality of bacterial genetics? Listen, if you consider this as a bacteria, okay, then this will be what? This is the chromosome of bacteria. This is called chromosome of bacteria. Okay, which is also known by the name of genophore nucleoid. Okay, this is chromosome of bacteria, and this is its plasmid. Okay, let me draw the plasmid in different color. Now listen, this is its plasmid. Clear? These are plasmids. Now listen, number of plasmid in a bacteria determines the what? Number of plasmid in a bacteria determines copy number. Okay, So in this bacteria there are two plasmids, that means copy number of this bacteria is equals to 2. Now listen, these are very important things you need to understand today. So copy number of this bacteria will be 2, right? Now listen, if I tell you, suppose this is a bacteria, suppose this is a plate, culture plate, and this is another culture plate. Clear? This is culture plate 1, this is culture plate 2. If I tell you that in culture plate 1 there are some bacteria, and in culture plate 2 there are also some bacteria. Now listen. Now if I tell you in culture plate 1 we have introduced an antibiotic known by the name of ampicillin, suppose, and in culture plate 2 we have introduced lactose. Clear? We have introduced lactose in culture plate 2, and in culture plate 1 we have introduced ampicillin, an antibiotic. Clear? Now my question is this, in which case do you think that chromosome will actively participate in metabolism? In antibiotic resistance or in lactose metabolism? If you don't know this, probably you will never understand this class. What do you mean? Listen, if you are thinking that what do you mean by this, that what is the importance of this lactose and ampicillin, now listen. Do you know in humans there are two kinds of nervous system? That is nervous system needed for day-to-day activities, right? That is central nervous system, peripheral nervous system. These are one category, and another category is useful for your stressful activities, right? That is sympathetic nervous system and parasympathetic nervous system, that is autonomic. Now listen, in bacteria also, bacteria doesn't have any nervous system, but it has got a special thing. What is that? That in stressful situation plasmids fire, and for day-to-day metabolism chromosome fire. Clear? Now listen, lactose metabolism, do you think it is stressful? Bacteria is taking glucose or bacteria is eating something, do you think it is stressful? Absolutely no, right? That means lactose metabolism will actually activate chromosome, but introduction of ampicillin creates a threat situation for the bacteria, right? Then plasmids will fire. This is plasmid, then only plasmids will fire. You must understand this very basic thing. Clear? So when ampicillin fires, when plasmid fires, when there is a threat to the bacteria. Clear? Clear? When there is a threat to the bacteria, ampicillin fires. Clear? When there is a threat to the bacteria, ampicillin fires, and when day-to-day metabolism is required, lactose fires. Chromosome fires. Clear? This should be crystal clear at first. This should be crystal clear. Clear? Now we are moving. So you now understand that chromosome is required for day-to-day metabolism and plasmid is required for emergency situations. Clear? This is one of the basic rule. Okay. Now listen. You must understand one thing, that if I tell you where are Lac Operon genes present? Lac Operon genes are responsible for lactose metabolism. That means what? Lac Operon genes are present in chromosome, not in plasmid. You must understand this very carefully. So the genes, beta galactosidase, yes, permease, I know you are more interested in bacterial genetics than human genetics, but for the time being you must concentrate here. That is, you must understand that Lac Operon genes are present in chromosome. Clear? Lac Operon genes are present in the chromosome of bacteria, not in plasmid. Clear? Okay, that's it. Now, very interesting insight, that is, do you know this term, partial diploids? Partial diploids or merodiploids? Partial diploid or metadiploid? Do you know this term? What do you mean by partial diploid or metadiploid? Now listen. You must understand that suppose this is a bacteria. Okay. This is a bacteria. And this is its what? Chromosome. This is its chromosome. Okay. And one thing I must tell you, that the chromosome is actually attached to the cell membrane of bacteria. Whenever a threat situation comes, then and then only the chromosome of bacteria actually gets detached from the cell membrane and they become an independent entity. These are plasmids. Okay. Now suppose this is your plasmid. This is not your plasmid. This is bacteria's plasmid. Okay. Now listen. Suppose this is your chromosome. Suppose this is the chromosome of bacteria and this is plasmid of bacteria. Now important thing you need to understand is this, that sometimes what happens is, due to recombination, plasmid and the chromosome, they join. That means what is the fate of this particular thing? Now listen. Suppose this is a bacteria now. This is having chromosome. This is what? This is the portion of the chromosome. This is the portion of the chromosome. This is the portion of the plasmid which is integrated into the chromosome. That means in some bacteria a funny thing happens, that is chromosome and plasmid, in some bacteria a very important funny thing happens, that is plasmid gets integrated into the chromosome. Okay. Now these bacteria in which the plasmid has been integrated into the into the chromosome, these type of bacteria are known as HFR strain of bacteria. HFR means high frequency recombination. High frequency recombination. Clear? We'll talk about these genetics, don't worry. High frequency recombination. This is HFR strain. Now listen with all your attention. Lac Operon was actually discovered by Jacob and Monod. Clear? Lac Operon was discovered by Jacob and Monod. And for their discovery, they were given a Nobel Prize in 1965. Okay, no problem in this. But in 1965, they were not the only scientist who were awarded the Nobel Prize. There was another person who was given Nobel Prize together with them, and many people don't know that. This person's name was Andrei Loew. He was Andrei Loew. Yes, why I am talking about this person, you know, is very important, because Andrei Loew discovered the transcription regulation in genes associated with virus. They discovered it in bacteria, Andrei Loew discovered it in virus. That's it. That's why they were given 1965 Nobel Prize in Physiology and Medicine. Clear? Now, they actually didn't discover Lac Operon, they also discovered the mutants. Okay, and they actually discovered how this thing actually works. Clear? Now, we will talk about the Jacob and Monod, model of Lac Operon. Clear? We'll talk about, we actually read Jacob and Monod's, model, which is really, really, important. Now, suppose this is a bacteria. Clear? Suppose this is a bacteria. Okay? This is suppose a culture plate. Suppose. Now, if you add glucose to this medium where this bacteria is placed, you will see that lactose metabolizing enzymes are going down. Concentration of lactose metabolizing enzymes are going down. It's very important for you to understand. Whenever you add glucose to a medium containing a bacteria, lactose metabolizing enzymes goes down. Concentration of lactose metabolizing enzyme goes down. Why? Because simple as, listen, biology is a really, interesting as well as, very much understanding and very much logical subject. You need to understand this right now with the logic, that whenever you add glucose, glucose is the preferred source of carbon in bacteria, because they can directly use glucose by applying glycolytic enzymes, right? But if you add lactose, they will have to at first break the lactose into glucose and galactose, and then they will have to convert galactose into glucose, and then they will apply the glycolytic enzyme on glucose. Now listen with all your attention. If you are giving directly glucose, if you are directly adding glucose, then you need to understand this portion, that is lactose metabolizing enzymes are not required. That's why their concentration was going down. But whenever you add lactose, the concentration of lactose metabolizing enzymes were going up. Now, there was a mystery, obviously, but important point you need to understand is this: why this is happening. Listen, in biology, the most important thing is why. Okay, now, if you think from a perspective of 1960 experimental model, you will understand the importance of this question. Suppose you are giving, glucose, concentration of these enzymes. How can you measure the concentration of these enzymes? It's very important. You just take the cells and you just break them with sonication or whatever you like. Just, take OD. You will understand the concentration, right? Now, they were seeing that whenever they add glucose, whenever they were adding glucose, lactose metabolizing enzymes, their concentration in the cell was going down. But whenever lactose was added, lactose metabolizing enzymes were, concentration of lactose metabolizing enzymes were going up. That is not important. The important factor they asked is, that is why in biology the most important question is why. They asked a very important question and found a solution. That's it. What was the question? The question was, how cell was able to distinguish between glucose and lactose? And the second question is this: how cell was able to sense which one is lactose and which one is glucose, and when to turn on these enzymes and when to turn off. That's it. These were the two questions they answered and got a Nobel Prize. That's it. Clear? We are today going to discuss these two answers, and you will learn Lac Operon. That's it. They actually, tried to explain this in their own way. If I can show you that in his name is Francois Jacob. Okay, Francois Jacob actually drew a diagram, okay, in his lab notebook, and if I can show you those diagrams, you will laugh at it. Why? That diagram was so important but so simple. That's why most of the researches going on around the world are so stupid you can understand. But the problem is this: they are not easy, they are simple. This was not an easy task, okay, but it was a simple task. That is, they can sense the lactose, that's why lactose metabolizing enzymes. But their question was how they can sense the lactose. That's it. What were the intracellular mechanism which was helping them? Why this question was around? Why this type of question came into their mind? Because at that time cell division was not discovered. Cell cycle also was, not discovered. That's why they wanted to know how an extracellular metabolic, thing, how an extracellular, signal can lead to change in intracellular mRNA concentration or enzyme concentration. That means this was the first thing they understood, that an extracellular thing is controlling, by entering into cell, intracellular genetic regulation. Why? Because lactose metabolizing enzymes are proteins. that is lactose, which is a non-protein thing, is controlling the what? Translation of proteins. How? That was the important question. Clear? Now, we have already discussed. Let's talk about the solution. Now listen. First things first, that what is the importance of Lac Operon in lactose metabolism? First question. What is the importance of Lac Operon in lactose metabolism? What do you mean by Lac Operon? I will talk about those later. You want to know what is Lac Operon right now? Okay, Lac Operon is nothing but control elements for structural genes. You have got your answer. Thank you. Okay, Lac Operon means structural genes, those genes associated with lactose metabolism, plus their controlling elements. That's it. So simple. But to understand this particular simple thing, you need to understand what is going on. Don't worry. In science, if you continue doing like this, then you will probably not get anywhere, because science is probably science doesn't depend on how much you know. Science depends on what you are doing with the knowledge. That's it. You may know many things, but you will not become a scientist in your whole life, because you don't know how to implement those things. That's important. Now listen. What is the importance of Lac Operon in this particular lactose metabolism? The importance of Lac Operon is this. Lac Operon is an area which is actually increasing the concentration of lactose metabolizing enzyme. That means in the Lac Operon, lactose metabolizing enzyme genes are present, and somehow they can sense the lactose and glucose. That's why their concentration goes up and down. Now listen with all your attention. Now you need to understand a basic thing. What is constitutive expression? Do you know what is constitutive mean? Constitutive expression. You know what is the meaning of this term constitutive expression? Do you know what is the meaning of this term constitutive expression? Constitutive expression means you want to try your luck. Constitutive expression means continuing expression. Okay, she's wrong, and you want to tell constitutive expression means associated with lactose. Completely wrong. Now listen, with respect to Lac Operon, I know that. Now listen. Constitutive expression means these type of, suppose if we consider a gene, and this gene's product is protein P. Suppose this is a gene, and this gene is ultimately after an mRNA. After an mRNA, it is giving a protein P. Clear? It is giving a protein P. Now you need to understand is this. This gene gives mRNA, and this mRNA is translated into a protein. Now listen. Suppose this is a cell, and hypothetically you have a door using which you can enter this cell. Now listen. If you enter 5 times, suppose this is a man standing there. Okay. This is a man standing there, and if this man enters this cell 5 times, probability is this: out of the 5 times, 4 times he will see this protein P is present, and it is synthesized. That means what? What do you mean by constitutive expression? Now listen, first you need to understand constitutive expression of what? Proteins or genes. Constitutive expression always occurs in case of protein, genes. From gene we get protein, that's why we always talk about constitutive expression of protein or genes. These are intermingled terms. Now listen. Whenever you talk about constitutive expression, we are talking about the constitutive expression of a gene. Constitutive expression means what? Constitutive expression means there is a tendency of this gene to produce protein at a continuous rate. Tendency. But you will always get this protein whenever you enter a cell. Whenever you will enter a cell, you will get lactose metabolizing enzymes. You understood my point? What I am saying is, lactose metabolizing enzymes show constitutive expression. That means whenever you enter a cell, you will get lactose metabolizing enzymes. But my question is, if there is no lactose, still lactose metabolizing enzyme will be present. Yes. They show constitutive expression means what? They show constitutive expression means whether lactose is present or not. To some extent, small amount of lactose metabolizing enzymes will always be present in bacteria. But my question is, what changes whenever lactose is introduced? My question is, what changes whenever you introduce lactose into the nutrient media? The change is the concentration of lactose metabolizing enzymes. That means these constitutive expression, listen, suppose a car is moving at suppose 10 kilometer per hour. Okay, now if you don't push your accelerator, then the car will move with a constant velocity of 10 kilometer per hour, right? Now my question is this, what is the change in acceleration? Zero, right? Now important point you need to understand is this, that this is the importance of constitutive expression. If you don't add lactose, change in expression will be zero. That means before 1 hour there was 40 microliter of enzyme, and after 1 hour there will be 40 microliters. Some has gone, some has degraded, but they are replaced by new proteins. But my question is this, whenever suppose you add lactose, what is happening? This 40 microliter is getting high and may reach up to 2000 microliter. Then that is important. This is an example, okay? Don't go for, because enzymes are not, calculated on the basis of microliter or something like that. Okay. Now listen. So whenever lactose is introduced in these cells, then, or in this media, lactose metabolizing enzymes expression goes high around 1000 times. That is important point you need to understand today. Now, clear? So Lac Operon genes show constitutive expression, but their expression reaches increases 1000 times more whenever lactose is introduced. Now, important point you need to understand here. Important point you need to understand here. Suppose this is a bacteria. This is its chromosome. This is plasmid. Right? This is chromosome, plasmid. Now, understand this very basic thing. Suppose this is the plasmid and this is chromosome, and if I magnify this portion, this is suppose Lac Operon. If I magnify this portion, okay, let's not magnify like this. Okay, now listen. Suppose this is your bacteria. Okay, suppose this is your bacteria's chromosome and you try to magnify this portion. Okay, so this is the magnified portion of your bacterial chromosome. Okay, this is the magnified portion of your bacterial chromosome. Now listen, with mapping experiments, Jacob and Monod found out that how the genes of lactose metabolizing enzymes are present, and they found out a very important and interesting thing, that is, suppose this is a DNA, portion of DNA, and these are the genes associated with lactose metabolism. There are actually 3 genes associated with lactose metabolism, 3 genes, that is LAC-Z, LAC-Y, LAC-A. LAC-Z, this gene produces an enzyme called beta-galactosidase, called beta-galactosidase, LAC-Y, Lac Y produces an enzyme called Lac permease. Lac A produces an enzyme called transacetylase. Clear? Now, why I am not drawing these portions? Now listen with all your attention. You know that every gene is controlled by a switch. Every gene is controlled by a switch. Those switches are called promoter. We have already discussed in transcription class in details. Now, this is the promoter, okay? The one which promotes transcription. This is the promoter, okay? It's very happy. Why you know? Because these genes are about to get transcribed. Now listen, this is a promoter. Clear? This is suppose a promoter. Now, the point you need to understand is this. Promoter is the switch of a gene. Now, you need to understand this very carefully, that promoter also has switches. That's it. Lac Operon actually proved that promoter also has switches, and if those switches are on, then only, if some switches are on and some switches are off, then only you will get the proper transcription of these genes. That means promoter is also controlled by some switches. That's it. This was actually the result of Lac Operon. So what are those areas which control the promoter? There is an area here which is called this is a very scary area. Why? Because this will actually inhibit transcription of these genes. Now listen, this area is called operator. This area is called operator. Clear? And that means operator is present downstream of promoter and upstream of these genes. Now listen, one thing you need to understand right now is these three genes together are called structural genes. These three genes taken together are called structural genes, and these are the control elements. Now listen, the third control element which is associated is this region of the DNA. This region is called cap region. What is cap region? Cap is actually an acronym for the real name of cap is catabolite activator protein. Clear? This is the name of cap. Okay, now listen. Point you need to understand is this, that cap region actually helps in transcription. How we will talk about that? Operator inhibits transcription and promoter is the area where RNA polymerase sits. Now listen, that means RNA polymerase will what? RNA polymerase will sit here together with sigma. We already discussed in transcription. What is the role of sigma? What is the role of RNA transcription? RNA polymerase. Now listen, RNA polymerase will sit in the promoter and they will try to move. RNA polymerase, suppose sitting with sigma, we will try to move here, but there is a inhibition in the operator. Clear? Now who is giving this inhibition? Now listen, operator is actually a region of DNA. How can a region of DNA inhibit an enzyme associated with DNA? It cannot. Listen, I am telling again. RNA polymerase is sitting in the promoter and it is moving towards the structural genes. Now, operator is actually what? A site. Operator is actually what? A specialized site in the DNA. Now, how can a DNA interact with a protein? It cannot. Now, my question is this: what is happening here? Now, listen with all your attention. There is a gene which is far, far away from all these things. This gene is called LAC. Clear? This gene creates a protein called repressor protein. Clear? Called repressor protein. This gene creates a protein called repressor protein. This repressor protein actually sits in the operator region, and this repressor protein actually blocks RNA polymerase. And due to the inhibition of this repressor protein, RNA polymerase will not move further or move forward. Therefore, transcription of all these genes are halted. Now listen, important point you need to understand here as well. That is, I have only introduced the genes and its control elements right now. Now, important point you need to understand is this. You know what is polycistronic mRNA? You know what is polycistronic mRNA? What is polycistronic mRNA? A polycistronic mRNA means an mRNA which is having many cistron. And cistron is what? Cistron is synonymous to genes. That means what? That means in case of bacteria, now listen, in case of bacteria, the structural genes produces a polycistronic mRNA. That means these three genes will never produce three mRNAs. They will produce a single mRNA, and these mRNA, this mRNA, sorry, I have drawn a DNA. Sorry, and this mRNA will have regions for LAC Z, LAC Y, and LAC A. Now listen with all your attention. Whenever, suppose this is an mRNA, okay, 5 prime, 3 prime. Now listen, whenever RNA polymerase, this should be DNA polymerase. Sorry. No, this should be RNA polymerase. Okay, sorry. Now, whenever translation is actually occurring, suppose this is your what? Small subunit of ribosome, large subunit of ribosome. Translation is occurring. Proteins are coming out from here, and this protein is beta galactosidase, and then after translation, these subunits fall off, and new subunits will attach here in the Stein-Dahlgren sequence of LAC Y. Again new ribosome will sit in the Stein-Dahlgren sequence here, and they will translate transacetylase. That means from a single mRNA you are getting 3 different proteins. This is called polycistronic mRNA. Clear? This is polycistronic mRNA. Okay, so we have discussed polycistronic mRNA and all these things, controlling elements, cap region. Okay, now listen. Now this is cap region, this is not cap, okay, this is cap region. That means what? In the cap region, cap protein will, or catabolite activator protein will sit. Clear? This is cap region. Okay. Now we were discussing this. Now important point you need to understand is this, that this repressor protein will sit in the operator region. Clear? Now when this repressor protein is sitting in the operator region, then RNA polymerase cannot move forward. Okay. Now, so this is actually the structure of Lac Operon. Now what is Lac Operon? Now what is Lac Operon, right? Lac Operon means structural genes plus these controlling elements. Okay, structural genes, that means Lac Z, Lac Y and Lac A, taken together with operator, promoter and cap region. These 6 portions taken together are called Lac Operon. Operon means gene together with its operating elements or controlling elements. Clear? Now we'll talk about the working of Lac Operon. We'll talk about the working of Lac Operon. Okay. Now, what happens? At first, we'll talk about Lac Operon functioning in a, in 2 different cases. That is, case 1 is when only glucose is present. When only glucose is present. Now, it's very easy for you to understand that when only glucose is present, the Lac Operon will be shut off. Lac Operon will be shut off, right? Now, okay. Listen. How this Lac Operon is shutting off, we'll try to discuss this. Now listen. The point you need to understand here is this, that there is an enzyme called adenylyl cyclase, the enzyme which you have read in your cell signaling classes, right? Now listen, it does this does the same job here. Okay, now listen, whenever glucose enters the cell, now listen, glucose inhibits a protein called 3-GLC. Glucose inhibits this 3-GLC. Clear? Now listen. Now this 3-GLC actually directly inhibits adenylyl cyclase. Clear? Now listen with all your attention. Whenever adenylyl cyclase is shut off, then ATP is not converted into cyclic AMP, right? Cyclic AMP, right? Now, when cyclic AMP is not present, then in the cap region, what happens is this is cap protein. Now listen, this is cap, okay, catabolite activator protein. Now listen, these cap proteins interact with cyclic AMP, and 4-cyclic AMP actually activate these 2 regions, and these cap proteins will sit in the cap region. These cap proteins will sit in the cap region. Now, when these cap proteins will sit in the cap region, then these cap proteins will actually activate RNA polymerase so that transcription can occur. Clear? Now listen. Suppose if you introduce glucose and glucose enters the cell, and this inhibits a protein called 3-GLC, and because 3-GLC is getting inhibition from glucose. Now listen, what is the important function of 3-GLC? 3-GLC actually, I was wrong, okay, no problem. 3-GLC actually what? Interacts with adenylyl cyclase and gives a positive response. Right? Now listen, when 3-GLC is getting inactivated, then this positive response is gone. Due to this, adenylyl cyclase will be shut off. No cyclic AMP, that means no cap protein activation. No cap protein activation means what? Cap protein will not now sit in the cap region. Now listen. So RNA polymerase is not getting an additional help from cap proteins or CAP, catabolite activator protein. Now listen. Now what about this repressor protein? Repressor protein has a constitutive expression. It is expressed all the time. Clear? Now whenever it is expressed all the time, that means it will sit in the operator region and it will have a negative impact on the RNA polymerase movement. It will not allow this RNA polymerase movement. That means whenever glucose concentration is high inside the cell, structural genes are shut off. This is the mechanism. Clear? Clear? This is how this whole thing actually works in presence of glucose. Now, case 2 is slightly different. Case 2 is when lactose is introduced in the cell. When lactose is introduced into the cell. Now listen. Before I start the lactose thing, you need to understand a very basic thing. That is, suppose this is suppose lactose. Lactose will enter the cell with the help of a protein called Lac permease. Lac permease. Okay, this is Lac permease. Now listen with all your attention possible. Lactose enters the cell with the help of Lac permease. That means I have already told that this Lac permease comes from obviously structural genes, obviously, but the point you need to understand is this: they have a constitutive expression. That means all the time some portion of Lac permease is already present in the cell. Now, when lactose enters the cell, lactose, due to constitutive expression of these structural genes, beta galactosidase will also be present. Now listen, what is the important role of beta galactosidase? Now listen, suppose this is, I'm not going into the details of all these things, but you need to understand now some very basic thing. Suppose this is your glucose and this is your galactose, and this whole thing is called lactose, right? Now listen, beta galactosidase actually acts here, clear? And it will cut this thing, and glucose is the preferred source, and glucose will be entering the glycolytic pathway. That's it. And this galactose will be converted into glucose, and then this glucose will be converted into pyruvate with the help of glycolytic pathway, and they will give rise to ATP and all these things. Now problem here is this: beta galactosidase sometimes work in a very much abnormal fashion. What is that abnormality? Now listen. Sometimes what happens is beta galactosidase actually, instead of working here, works in a very much abnormal fashion. That is, it actually does this. This was what? This is 1, 2, 3, 4, 5, 6. 1, 2, 3, 4, 5, 6. Now listen. This was 1,4 glycosidic linkage. Now listen. Instead of breaking this 1,4 galactoside, this is beta 1,4. Okay. Now listen. Instead of breaking this thing, beta galactoside sometimes what it does is, it actually changes the position of this linkage and it creates beta 1,6 linkage. Okay, and this type of lactose where beta 1,6 is present is known as allolactose. This is known as allolactose. Okay, This is called allolactose. That means whenever, suppose there are 10 lactose entering the cell, suppose there are 10 lactose entering the cell, at least 2 will be converted into allolactose and other will be converted into glucose and galactose. That means there is always formation of allolactose whenever lactose enters the cell. Now you need to understand very carefully now. Listen. Many persons don't understand how allolactose actually, what is the role of allolactose? Can anyone tell? Whoever is seeing this video right now, they should pause and think about this, that what is the important role of allolactose in Lac Operon? You know that it actually inhibits what? Repressor protein. How? Can anyone answer this? How? How allolactose interacts with repressor protein and it inhibits repressor protein? If you can't answer this thing, that means you probably don't know the structure of, repressor protein. It's very important for you people to understand that what is the structure of repressor protein. Listen with all your attention. Repressor protein is actually not a single protein. You need to understand this first. Repressor protein is not a single protein. It's not a single protein. It's a tetramer like hemoglobin. It is actually 4 polypeptides coming together to form a single protein. That means it has got what? Tertiary structure. It has got tertiary structure. That means suppose this is protein 1, protein 2, protein 3, protein 4. These 4 proteins taken together is called repressor protein. These 4 proteins taken together is called repressor protein. Now listen. Allolactose actually binds with one of these polypeptides and the whole structure of repressor protein is destroyed. And therefore, they are unable to interact with the DNA of operator region. Therefore, they are unable to interact with the DNA of operator region and therefore they are unable to bind operator region. That's it. Clear? Clear? Okay. That means what allolactose does is allolactose interacts with tetrameric repressor protein. It's a tetrameric, right? It's a tetrameric repressor protein. Allolactose inhibits this repressor protein. Clear? With the inhibition of this repressor protein, there is no inhibition of RNA polymerase, right? There is no inhibition of RNA polymerase. Now listen. When glucose was entering the cell, there was this inhibition of 3-GLC, right? Now, whenever glucose is absent, there is no such inhibition, right? There is no such inhibition. As lactose is entering the cell, there is no such inhibition. Because of this 3-GLC will now activate what? This 3-GLC will now directly activate what? Adenylyl cyclase. And adenylyl cyclase will convert ATP into cyclic AMP. And this cyclic AMP will now sit in the cap region, in the head of what? Catabolite activator proteins, these proteins. And these proteins will sit in the cap region. And this will initiate, this cap proteins or this CAP or this catabolite activator, let me talk about cap proteins. I will, give this name as cap proteins, a new name, suppose. Okay. I can't always say catabolite activator proteins. Okay, now listen. Suppose these cap proteins will now sit in the cap region and they will initiate, then they will help in the binding of RNA polymerase properly to the promoter. Now listen, with the cap proteins placed in the cap region and no protein or no other inhibitory factors, these RNA polymerase will move forward and will give rise to what? Beta galactosidase, lac permease and transacetase. Now, what is the role of beta galactosidase? You know. What is the role of lac permease? You know. What is the role of transacetase? Many people say acetylates and glucose, acetylate lactose, but there are still some argument with respect to the functioning of this transacetase. Okay. Okay. So these are the two cases of functioning of Lac Operon. When glucose is present, what happens to adenylyl cyclase? Clear? And when lactose is present, what happens to adenylyl cyclase? And what is the role of adenylyl cyclase in the activation of cap protein? Clear? Or catabolite activator protein? Clear? That's it. Now, you have learned only how Lac Operon works. That's it. That means an extracellular signal is coming into the cell and telling the cell which genes to turn on and off, right? This is Lac Operon. That means extracellular signal controlling the gene transcription. That's it. But my question is this, now we'll talk about some mutations. Now we'll talk about mutations in laci and these operator regions. Why these only two? Because in most of the cases, in many bacteria, these two regions are more susceptible to mutations. And these mutations give rise to many different types of phenotypes which are abnormal. Okay. Right now we'll take some break for some time, then we'll come back, okay, and we'll talk about the mutations and we'll discuss the sums as well.